Let's take a look at a recently discovered archaic Homo sapien, a child from Denisova Cave that has been dated as a contemporary of the Neanderthals. In 2008, a new hominid was discovered in Siberia that proved to be a contemporary of Neanderthals and early modern humans, contributing to our understanding of archaic Homo sapiens in Eurasia. The stratum containing the fossils, a piece of a child's finger bone and 2 molars, was dated to approximately 50,000 to 30,000 years ago. Because so few fossils were found, it was uncertain which species the material belonged to, but it was expected to represent either Neanderthals or early modern humans, both of which inhabited Denisova Cave at various periods since 200,000 years before present, and were present in Eurasia at the time the fossils were deposited. Given the limited fossil finds, anthropologists used ancient DNA, ADNA, to study the materials. The cave remains at freezing temperatures year-round, which helps preserve the ADNA and inhibited bacterial activity that would contaminate it. The researchers began analyzing by sequencing the mitochondrial DNA code of the finger bone and molar, followed by a small portion of the nuclear DNA code from the finger bone. Because of the optimal conditions at Denisova Cave, the researchers were able to recover approximately 70% of the DNA of what proved to be a young girl. Much better results than the 5% that is usually recovered from ancient bones. Mitochondrial DNA showed that the individual from Denisova Cave represents a new, separate branch of hominid. The preliminary analysis suggested that although Neanderthals may have contributed 1 to 4% of the DNA of non-African humans today, the new archaic Homo appeared to have contributed even less, with one exception: modern human populations in Malaysia, where the new archaic Homo DNA might contribute as much as 3 to 6% of the genome. This evidence was preliminary, as not much ADNA was sequenced. Ancient DNA is fragmentary and fragile, and difficult and expensive to work with. Previously, ADNA methods utilized double-strand DNA only, and the fragmentary single strands remained unsequenced. In August of 2012, the same research team published the complete nuclear DNA code for the Denisova Cave hominids. This was only possible through new DNA sequencing technology developed by Mathis Meyer, involving the application of single strands of DNA rather than intact double helices. Researchers have been attempting this method for years with low success rates, and its successful application in 2012 was a huge step forward for ADNA studies. In addition to 99.9% of the genome that was copied at least once, 92% of the ADNA was sequenced to more than 20 copies. This is better coverage than what was obtained for the first human genome sequence, the first Neanderthal genome sequence, and the earlier efforts with the same fossils in 2010. The new method provides potentially new understanding of the phylogenetic position of the individuals from Denisova Cave, as well as some clues as to their physical appearance and likely behavior. The early report of DNA showed a very ancient branch for the individuals from Denisova Cave off the human lineage at around 1 million years ago, which would imply that hominids may have left Africa in an early migration prior to that associated with the Neanderthal and human lineage. The new DNA data shows that Denisova individuals are more closely related to Neanderthals than either is to us. That is, the group represented by the Denisova Cave individuals and Neanderthals diverged from each other after our split from the Neanderthal lineage. The date of the split between Denisova Cave hominids and humans is now placed between 700,000 and 170,000 years ago, a wide range that reflects uncertainty over mutation rates to use in the context of the molecular clock. The new technique used to sequence the DNA permitted a new date range to be obtained for the fossils using the molecular clock of 82,000 to 74,000 years ago. This date range appears to be too old given the archaeological context, and researchers acknowledge that we may be assuming too rapid a rate of neutral mutation in the DNA. However, the study shows how in the future aDNA may be useful in assessing ages of fossils, where other dating techniques are unusable. The new technology used in the 2012 study will also facilitate analysis of more highly degraded DNA from more ancient hominids. The genetic diversity of the Denisova cave individuals was low, indicating population sizes may have been small. Apparently, while human populations were rapidly expanding around 250,000 to 125,000 years ago, the populations represented by the Denisova individuals and of Neanderthals were shrinking. It may be that genetic adaptation among early modern humans contributed to our ancestors' success. But what are these human Denisova hominid differences? Features unique to humans that are not shared by the hominids from Denisova cave include 111,812 single nucleotide changes, at least 367 of which would have phenotypic effects, and 9,499 insertions and deletions. Some of the expressions of these changes are known. These include changes affecting brain functioning, not size, and nervous system development, including changes implicated in autism and language disorders. Other changes may have affected language, like the FOXP2 gene. 34 changes unique to the human genome are associated with human diseases, including some affecting the skin and the eyes, suggesting skin and eye physiology changed recently in our evolutionary past. In terms of physical appearance, the girl from Denisova cave whose DNA was sequenced for the study probably had brown hair, eyes, and skin. Only the sequencing of more DNA from other individuals will reveal whether these traits are common among other members of this group. Interestingly, the Denisova cave individual appeared to have had 23 chromosomes, with chromosome 2 representing a fusion of chromosomes that in great apes are separate. Evidently, our 23 chromosome karyotype developed sometime before our split with the gene pool of the Denisova cave individuals. The discovery that this new archaic hominid's DNA is present in modern Malaysian humans is interesting. There is a large gap between Siberian and Malaysians where modern humans show no traces of DNA from Denisova cave gene pools. Researchers do not think that new hominids traveled to Papua New Guinea. Rather, they suspect that they were once widespread throughout most of eastern Eurasia, much as Neanderthals were once widespread throughout western Eurasia. Southern groups likely encountered Malaysian ancestors and interbred with them, after which those Malaysian ancestors moved with their archaic Homo sapien DNA to Malaysia. Additionally, the researchers discovered that among Papuans, the new DNA is more often found on autosomes rather than on the X chromosome inherited from the mother, suggesting interbreeding may have involved males of the new hominid variety breeding with early modern human females. This is the first time a new hominid has been identified chiefly on the basis of genes rather than physical features. This complicates how we situate this new variety of archaic Homo within our framework of knowledge concerning hominid evolution that is solidly and necessarily built on morphological evidence of species designations. The researchers expect to find other groups of archaic humans using the new sequencing techniques. In the future, expression studies must be done, which establish what exactly the Denisova cave individuals and human genes do, that is, linking genotypes to phenotypes. Although the fossils are scarce, DNA evidence tells us that a previously unknown type of archaic Homo once populated parts of Eurasia. Hopefully, additional fossils from this group will be identified elsewhere in Asia to further our understanding of archaic Homo sapiens and our evolutionary past.